First and foremost, I'd like to thank the organizers for the invitation, and for giving me the opportunity to discuss two rivaling single molecule, single cell spatial transcriptomic platforms that are out there and currently being used to understand RNA localization and dynamics, both in tissue development and in disease. As you guys all know, single RNA detection, gene expression has been analyzed using various different approaches, where you're looking at a single gene using either northern blots or qPCR approach. However, these approaches are low throughput and you're detecting and following a single gene event. And it wasn't until bulk sequencing technology was developed and where you're able to go from a single gene to thousands of genes. But this really relied on the utilization of a heterogeneous cell mixture or tissue. And in the most recent years, we've seen a development of a number of single cell RNA sequencing approaches, which allows you to analyze thousands of cells and thousands of transcripts per individual cell. One of the caveat, though, or limitation of single cell RNA technology is that you lose spatial and positional information within the cell. And in order to recapture this information, people have been using single molecule in situ hybridization, or SMFISH. And what this technology really relies on is the utilization of complementary antisense probes that are designed to a particular mRNA of interest that are actually fluorescently conjugated. And what you typically have is about 30 probes per transcripts. And what this allows is to enhances the signal to noise ratio of a particular transcript with a cell. You could identify the location of RNA within the cell and identify precise copy number information. However, this approach is very limited, and it's actually limited by the colors, and it's actually very costly to design SMFISH probes. So how can one use a limited number of colors to actually detect a large number of genes? And so one strategy people have used is the serial hybridization or sequential hybridization, where you have three SMFISH probes hybridizing at the same time, each with a different, labeled with a different color. The tissue is imaged, and then you strip the probe and rehybridize this multiple times. This is a great strategy, but analyzing a transcriptome or a large number of genes is complicated, because this would require about 7,000 hybridizations to actually take place to cover about 20,000 genes, which is the number of transcripts that is expected in the transcriptome. So to overcome some of these challenges and bypass the 7,000 rounds of hybridization, there's two technologies out there that are have used multiplex molecular barcoding on SMFISH probes that allows basically the detection of thousands of genes within thousands of cells. And this is known as MERFISH, which is shown here. And this was developed by Xiaowei Zhong's lab, as well as SeqFISH that detects either introns or exons. And this was developed by Long Kai's group. They're both theoretically very similar in the design. However, the only major difference between the two approaches is the way the genes are actually being called out, or the molecular barcoding schema readout. And I'll walk you through this briefly, but what you typically do is actually design probes either to exons or to introns of the transcript of interest, and it doesn't really matter which one you actually choose. If you design probes against exons, of course, this creates molecular crowding issues that you need to actually consider. So thinking about the types of transcripts and the number of hybridizations that you actually want to consider. But each individual RNA is tiled with about 35 to 45 oligonucleotides, and these oligonucleotides have actually it's very similar to the SMFISH. They have a complementary probe region here that is complementary to the RNA, and you have 4 different molecular barcodes that you actually attach to each oligonucleotide. And again, you have 35 to 45 probes per gene, depending on the approach that you're actually using. And these these oligonucleotides have 5 molecular barcodes that actually define a gene that are scattered onto these 35 to 45 probes used together for an RNA. And so then in order to read out these barcodes in the cell, you actually have these readout probes that are fluorescently conjugated and are complementary to the molecular barcode. So in each round of hybridization, you will have 3 molecular barcodes that are being used with different colors to read out different transcripts per cell. And so just to briefly walk you through this, in seafish, you're doing around 20 rounds of hybridization. In merfish, you're doing in seafish, you're doing 20 rounds of hybridization, and each of the hybridization runs has three readout probes that are analyzed at once. And this is usually the readout probes are using three color readout probes. So you have 594, 488, and 647. So, and theoretically, you're actually collecting 60 data frames per cell. 60 data frames per cell. If you're actually analyzing exons, they needed to increase the number of rounds of hybridization because of the expansion microscopy and to avoid crowding. And in this case, you're collecting 240 data frames per cell. The theoretical maximum number of genes that can be detected using the seq-fish approach is 12 to the raised to the power of 5. 12 is the number of pseudocolors that are actually used per barcoding round. And 5 is the number of barcoding rounds used in total. So these 20 hybridizations are divided across 5 barcoding rounds. And I'll walk you through this a little bit later. So the theoretical hypothetical maximum number of genes is 248,000 per cell, and you actually need to detect the gene needs to be observed 5 times in the 60 data frames, but 4 times is actually sufficient to call a transcript. Merfish is actually very similar to seafish, and it uses 23 rounds of hybridization. Again, you're using 3 colors, so you're collecting 69 data frames per cell. The calling mechanism for a gene is a little bit different. Within the 69 data frames, you're scoring whether an RNA is present or absent in these different rounds of hybridization. So you're using a binary coding system. And so the number of theoretical genes is 2 raised to the power of 23, which is the number of hybridizations that are actually used. And again, this approach relies on detecting the gene 4 times out of the 69 different hybridizations. So how do you actually get 12 pseudocolors in the seq-fish approach? And this is really, it's just a schematic of how the 12 pseudocolors are defined. So within a barcoding round, you actually have 4 different rounds of hybridization. And as I said, each round of hybridization has 3 colors. So this is 594, 647, and 488 that is hybridized. And then you image, strip, reprobe, so on and so forth for each of the different rounds of hybridization. So each barcoding round has 4 hybridizations, and this has a total of 12 pseudocolors, and this is what is used. A gene is only detected once per round of barcoding. And so the way you actually end up calling out a gene is by doing 5 different molecular barcoding rounds. And as you can see here, in barcoding round 1, a particular gene is going to be here. You're picking out a particular gene 4 using barcode 4. In the second round of hybridization, the same gene in the same position within the cell is being detected by barcode 9. In the third round of hybridization, as you can see, this is detected by barcode 2 and 12 and so on and so forth. And by using this combination of barcodes, you could actually deconvolve what this particular gene is in the cell. And so this is the data frames that you actually get. So you get 12 data frames from the 12 pseudocolors multiplied by 5 rounds of barcoding. And so what you're expecting to see is that a gene would appear 5 times across the different barcoding rounds. So in merfish, this is actually very similar, but you don't use pseudocolors, and instead you use 23 rounds of hybridization with 3 fluorescent probes. And again, here the only difference is that you're not using the pseudocolors or barcoding rounds, and here you're just calling the presence or absence of the signal in 69 data frames, and you need 4 to be present out of the 69. So using this technology, we wanted to understand the spermatogenesis process and germ cell differentiation process. And as you can see, the testes is a bag of tubules, and within a cross section of the tubule, there's multiple germ cell types that are actually organized where the stem cells are at the basement membrane and the sperm is towards the lumen. We've used single cell RNA-seq to define the cell types across in multiple species. And by using iterative clustering, we're able to define subtypes for the different cell types, the major subtypes, as well heterogeneity within a particular subtype, which really led us to the question is that where's this heterogeneity coming from? Is it coming from positional information within the cell or across the different stages of the seminiferous epithelium? So to do this, we wanted to apply these technologies, and what we used is a combination of merfish and seekfish, and we used a small number of genes initially to map across this germ cell differentiation trajectory. And so we've selected 80 genes that are actually kind of marked from spermatogonia to elongating spermatids. And this work was done by Gabe Mansky and Chen Ying, a graduate student in the lab, as well as with help with Morgan Anaka from Arvind Rao's lab. And using the combination of merfish and seekfish, we were able to detect 76 out of the 80 genes that we had initially set out to identify. And using our in-house spatial data browser, we were able to map the genes across the seminiferous tubule and also reconstruct the single-cell RNA-seq clustering information as well to get more information. And with that, I just want to say thank again the students that have actually done the work. And this is done by two really talented graduate students, Gabe Mansky and Shen Ying and Morgan Anaka. And this is done in collaboration with June Lee and Arvind Rao's group. Thanks.